Now the time to start is over. So let's start this session. This session is MAFLD3, is a MAFLD, basic understanding. 4 speakers are planned, but the first speaker, Shinji Tanaka, will present on video or web. So we changed to the first speaker to Professor Takumi Kawaguchi. I introduced Takumi Kawaguchi. Takumi Kawaguchi is a well-known hepatologist in MAFLD in Japan. He is a professor of the Department of Molecular Oncology, Department of Hepatogastroenterology in Kurume University. And his area is static liver disease or hepatocellular carcinoma. So the title of this session connected these to his theme. Okay, and today's title is Role of SGLT2 inhibitor in MAFLD-related HCC. Professor Takami, please.
Thank you, Professor Ito and Hikita, for your kind introduction. My name is Kawaguchi from Kurume University of Medicine, and today I'd like to talk about the role of SGLT2 inhibitor in the MAFLD-related HCC. Here's my COI. In the guideline of an evidence-based clinical practice for MAFLD-NASH 2020 in Japan, there is one clinical question regarding to the SGLT2 inhibitor. The question is SGLT2 inhibitor is effective for NAFLD or NASH? The answer is, in NAFLD NASH patient with type 2 diabetes, SGLT2 inhibitor improves liver enzymes and histological findings, and its administration is therefore suggested. However, at that time, the evidence is still limited, so the evidence level is C and strength 2. However, after that, there is a lots of evidence has reported, and then today I summarize in terms of the hepatic steatosis, fibrosis, as well as HCC and prognosis. This is the study of a single-arm trial conducted by Professor Sumida using the lucioglyphrogen is on an SGLT2 inhibitor. They found that the significant reduction of serum level of ALT and gamma GDP was seen during the time course. And then what is interesting is the primary outcome. He evaluated hepatic steatosis using the MRI-PDFF, which is the most accumulate evaluation method. And then he found that after 24 weeks of treatment of lucioglyphrogen and an SGLT2 inhibitor, there is a significant reduction of hepatic fat contents evaluated by MRI. The reason why SGLT2 improved hepatic steatosis is something like that. SGLT2 inhibitor promotes the urinary excretion of the glucose that reduces the reduction of visceral fat and leading to the improvement of hepatic steatosis. But I was wondering if the SGLT2 inhibitor directly affects to the hepatocyte and the liver, and then we investigated the expression of SGLT2 in the liver. What we found is the liver express the SGLT2. The left panel is a control using the kidney, and the SGLT2 occurs in proximal tubes, as reported before. And what we found is the expression of SGLT2 was seen in hepatocytes. You can see the brown dots in the hepatocyte. In addition, expression of SGLT2 was seen in the bile duct, here as like in the surface of the bile duct. So SGLT2 occurred in the kidney as well as the liver. The similar findings has been also reported by the Chen et al., and which is in the metabolism in 2023. Compared to the normal liver, even in steatosis, the SGLT2 inhibitor is upregulated, and the upregulation is further marked in the patient with NASH. The corresponding messenger RNA expression is similar. The expression level was increased in the patient with steatosis and further increased in the patient with NASH. Then I was wondering how SGLT2 inhibitor improves the hepatic steatosis directly to the hepatocyte. The answer is reported that the Frontier Pharmacology in 2021, SGLT2 inhibits the SGLT2, which is located in the surface of the hepatocytes, and it activates the AMP kinase and mTOR pathway, which causes an induction of autophagy. You know that autophagy is the degradation of many things. So the induction of autophagy improves the hepatic steatosis through the autophagy. Then I'd like to move on the theme on the hepatic fibrosis. The biggest problem on the hepatic fibrosis is lack of sufficient evidence, especially there is no meta-analysis to investigate these beneficial effects of SGLT2 inhibitor. So I performed a pooled meta-analysis using 5 phase 3 controlled trials conducted in Japan. The total number of the 5 phase 3 clinical trials was the 1,310. We excluded a patient with 23 diseases, which may affect the contents of hepatic steatosis as well as the hepatic fibrosis. The remaining 634 patients were used to the analysis. The analysis was performed by the propensity score analysis by the inverse probability of treatment weighting, IPTW, using the listed here 19 covariates. The total analyzed subject is 493, and the lucioglyphosine includes 302, and placebo includes 191. The primary outcomes is changes hepatic steatosis and hepatic fibrosis indexes. Secondary outcome is changes cardiometabolic dysfunction. Here are the patient characteristics as the background. Since this is the phase 3 clinical trials using the lucioglyphosine and an anti-diabetic medication, so that the age and sex and BMI and hemoglobin A1C is similar. There's no significant difference between the two groups, especially the hemoglobin A1C is 7.9 to versus 7.8. Fatty liver index, APRI, CLMV level, and CMLT level. For these markers, there is no significant difference between these two groups. This is a result of the primary outcome. After the treatment of lucioglyphosine as the SGLT2 inhibitor, there is a significant reduction in fatty liver index, 12 and 24 weeks. The similar findings were observed in the MAFLD liver fat score as well as hepatic steatosis indexes. So the lucioglyphosine significantly improved various indexes for hepatic steatosis compared to placebo. Here is another primary outcomes. The indexes for hepatic fibrosis. This is a stratification analysis according to the FIB4 index greater than 1.3. In these groups, there is a significant reduction in HepaMet fibrosis score, which is the most sensitive marker for greater than F2. That is shows a significant reduction at 24 weeks after the treatment. The reduction was also seen in APRI at the 24 weeks. So that HepaMet fibrosis score and APRI significant improvement was observed in these markers and in the lucioglyphosine groups compared to the control groups. As far as I searched the literature, I found that that there is an interesting study reported that effects of SGLT2 inhibitor on the hepatic stem cells. The report by Shan et al. And published its metabolism in last year. They found that SGLT2 affect to directly to the hepatic stem cell and downregulate microRNA 34A/5P, and then that upregulate of germ2. That causes inhibition of TGF-beta and leading to the improvement of hepatic fibrosis. So these data indicates the direct effects of SGLT2 inhibitor to the hepatic stem cell and then hepatic fibrosis. Another important findings was published by the Professor Takahashi from Saga University. They conducted randomized control trial versus SGLT2 inhibitor versus control. The control is not a placebo, the other anti-diabetic medication rather than the SGLT2 inhibitor. The study period was 72 weeks. Also, there is no significant difference in steatosis and inflammation between the two groups. Significant improvement was seen in ballooning. SGLT2 inhibitor improved 50% of the ballooning. And an SGLT2 inhibitor, approximately 60% of patients shows an improvement of fibrosis, which is quite significant versus control. That is only 60%. This is a result of secondary outcomes. And since this is an anti-diabetic medication, significant reduction was seen in hemoglobin A1C and a HOMA-IL value, which is index for insulin resistance. In addition, this marker, significant improvement was seen in HDL cholesterol and uric acid. So the ruxoligliflozin improved the various cardiometabolic dysfunction compared to placebo. And finally, I'd like to talk about the HCC and the prognosis. As far as I researched the placebo, there is some reports investigating the effects of SGLT2 inhibitor on the HCC in animal model. Obara and Professor Shimizu from Gifu University reported that the preventive effects of SGLT2 inhibitor dapagliflozin on the tumor genesis in obese and diabetic mice. Then Shiba and Ogawa from Kyushu University reported that canagliflozin is also an SGLT2 inhibitor, attenuates the development of SCC in a mouse model of NASH. Furthermore, Kaji and Yoshiji from Nara Prefecture University investigated the effects of SGLT2 inhibitor on the liver cancer cell growth and the angiogenic activity. Here is a result from the Kaji and Yoshiji et al. They shows that the canagliflozin significantly suppress the cell proliferation of HU7 cells and hepatitis cells. Similarly, SGLT2 inhibitors significantly suppress the tumor volume of the HU7 cell and hepatitis cells. We also investigate the effects of SGLT2 inhibitor on HCC progression and found that there is a significant reduction of number of HCC and volume of HCC as like here. But I was wondering if it's the direct effect of SGLT2 inhibitor on the HCC. Then we investigated the first expression of SGLT2 on the HCC cell line, and we used the 4 different antibody for SGLT2 and found that any antibody, all of antibody reacts to the HEP3B and HU7 cells. And then by the support of the Professor Yano from the pathology department of Columbia University School of Medicine, we got tons of HCC cell lines and investigated the effects and the expression of SGLT2 and found that all of cell lines express SGLT2. All of them are on the human HCC cell line. Then we investigated the localization of SGLT2 in an HCC cell and found that SGLT2 co-localized with the mitochondrial marker. In the study, we colored SGLT2 by red and a mitochondrial by green, so that in overlay image, you can see here yellow dots. That means the co-localization of green and red equal yellow. The similar find was also seen in the cell fraction examination. In the mitochondrial fraction, there is a very strong expression of SGLT2 in HEP3B cell and Q7 cells, but the weak expression was cytoplasmic. So that these data indicated that SGLT2 is localized in the mitochondria in HCC cells. Then we investigated the SGLT2 inhibitor on the proliferation of HCC cell line. We adding to the SGLT2 inhibitor on the culture medium of the HEP3B cell and Q7 cells. And the concentration we used here is a physiological level, and then SGLT2 inhibitor significantly suppress the cell proliferation by the dose-dependent fashion in HEP3B cells and Q7 cells. Then we investigated how SGLT2 inhibitor suppress the cell proliferation in HCC by using the metabolic analysis and proteomic analysis by using a two multi-omics analysis. We summarize the data in the single maps here. And blue line shows a downregulated pathway, and then blue dots shows that downregulated metabolites. In contrast, red line shows the upregulated pathway, and then red dots is upregulated metabolites. By indicating that the altered metabolic pathway and metabolites, we found that there is some points in the SGLT2 effect. The one is the beta oxidation, it's in the mitochondria. The another one is oxidative phosphorylation, which is also in the mitochondria. So we summarize these data in the cartoon as like here. This is the our proposed pathway, like SGLT2 inhibitor, how summarize and how inhibits the cell proliferation of HCC cells. What I think is the SGLT2 inhibitor suppress the SGLT2, which located in the mitochondria is HCC, that causes the reduction of ATP production, so that the ATP decrease. However, the cancer cell have to be survived. So how do they do that? They probably do that activates the beta oxidation that causes the overproduction of ketone body. As you know, ketone body is one of the suppressor of cell proliferation, so that the activation of beta oxidation is one of the pathway that SGLT2 causes cell proliferation. The another pathway is activation of AMP kinase that is stabilize the cell through the fatty acid synthesis. Another interesting point is the SGLT2 inhibitor prevents the PD-L1 degradation. So the SGLT2 in the cancer cell prevents the degradation of PD-L1 that causes the against or protect from the attack by the CD8 positive T cell. However, SGLT2 inhibitor allow the cancer cell to degrade PD-L1 that cause the attack or allow the attacking by the CD8 positive T cell, that leading to the suppression of the tumor. Here I summarize the role of SGLT2 inhibitor in MAFLD-related HCC. The first point is hepatic steatosis, then next is fibrosis, and finally I show the effects of SGLT2 inhibitor on HCC and prognosis. And here is my colleagues, and they work harder, and I like my sincere thanks all of the colleagues. Thank you, and thanks for your attention.
Thank you, Professor Kawaguchi. And Professor Kawaguchi talked about many aspects of the SGLT2 inhibitor. One is the interference of the steatosis, and one is the interruption of the fibrosis. One is stop the replication of HCC cells by several pathways. In this time, the audience who want to confirm some result of the Professor Kawaguchi, please raise your hands.
Stand in front of the mic.
Okay. No special confirmation is needed. Okay. Okay. Thank you, Professor Kawaguchi.
Thank you.
And all the discussion will be made after all the speakers finished. Okay.
So let's move to the next session, next speakers. So next speaker is So next speaker is Vincent Wang, please. So Vincent Wang is the professor and head of the Division of Gastroenterology and Hepatology and Department of Medicine and Therapeutics of Chinese University of Hong Kong. And his title is the role of intestinal TM6SF2 in MAFLD. So please, Dr. Wang, please.
Thank you very much for your kind introduction. Good afternoon, ladies and gentlemen. First of all, I'd like to thank the organizers for the opportunity to speak today. Here are my disclosures, and none of these would be related to the animal work. So before I start my talk, I would like to highlight that this is really a group effort of the entire State Key Laboratory of Digestive Disease at our university. So in particular, Dr. Jennifer Chang is responsible for most of the laboratory work. I would also like to highlight that Professor Hee-Won Lee from Korea actually started the project, but because it lasted 7 years, so many colleagues were involved. So what was the story about? So 10 years ago in Nature Genetics, we have identified the second gene polymorphism that is associated with the development of fatty liver disease. So this was our exon-wide association study. So as you can see, PNPLA3, the first gene identified to be robustly associated with fatty liver, is here. But you can also see that TM6SF2 is also highlighted here in this Manhattan plot. And since then, the association between the gene polymorphism with fatty liver disease has been confirmed by multiple cohort studies, including some from Asia and one from our group. So either if you analyze the data by dichotomizing the patient as having fatty liver or not, or using liver fat content as a continuous variable, you see a robust association. Okay, so in the past 10 years, actually, many people have already worked out the function of TM6SF2 in hepatocytes. So like many of the genes that are responsible for fatty liver development, they are proteins present on the lipid droplets inside liver cells, and TM6SF2 is no exception. So we know that it is on the lipid droplets, and it is responsible for VLDL lipidation and release. So inside hepatocytes, the VLDL would be packaged with lipid added, and then finally going through the Golgi process and then released into the circulation. So this is what is happening. If TM6SF2 is normal, then the mature VLDL would be released in the circulation. In contrast, if you have a mutation in the TM6SF2 gene, or if in animal models or cell line models, if you suppress the expression of TM6SF2, what happens is that the lipid is no longer released in the form of VLDL. In contrast, it will be retained inside the liver, resulting in hepatic steatosis. This is the bit that we have already known inside the liver. However, if you look at the data more closely, you would realize one very interesting phenomenon. So let's look at the expression of TM6SF2 in different tissues. you would realize that kidney is here, liver is here, definitely important. However, the highest expression of TM6SF2 turns out to be in the small intestines in both humans and animals. So the logical question is, what is TM6SF2 doing in the intestine and whether it contributes to fatty liver disease? So it is already 7 years ago when we looked into our liver biopsy cohort, so for patients with biopsy-proven MAFLD. So we demonstrated for people homozygous for the TM6SF2 minor allele, you see one interesting phenomenon, that is the lipopolysaccharide binding protein, the biomarker for endotoxemia is grossly elevated. If anything, it doubles if the patient is carrying the mutant protein. So this led us to ask several important questions. So does intestinal TM6SF2 contribute to the development of fatty liver disease, or more importantly, inflammation and fibrosis? And if so, what is the mechanism? And whenever we talk about the gut, of course, gut permeability and dysbiosis would be something we have to consider. And finally, for clinicians in the audience, of course, the logical question is whether the detrimental effect can be mitigated. So first, we generated global knockout mice for TM6SF2. So these animals do not express TM6SF2 in any tissue anymore. So like what other groups have demonstrated, when we fed the animals with high-fat, high-cholesterol diet or choline-deficient high-fat diet, you can see a marked increase in the degree of hepatic steatosis. So a lot of fat, and also there is inflammation inside. So everything increased. So steatosis, inflammation, the nephrophytosis, nephroactivity score, as well as fibrosis. So this is what we have already known. What is interesting is we started to generate organ-specific knockout animals. So first we had liver-specific knockout mice. So now TM6SF2 is no longer expressed in the liver, but it is expressed in other organs. So again, we used the same experimental design and fed the animals with the same diet. This time we saw something differently. So you see the steatosis is still there. In fact, the degree is no different from the global knockout mice. What is different, however, is we do not see a significant increase in inflammation, and there was no fibrosis either. So this would be indirect evidence suggesting that if you do not have the contribution of the gut, if you only suppress the expression of TM6SF2 in the liver, you only increase fat accumulation, but you do not induce inflammation. And when we fed wild-type mice with a choline-deficient high-fat diet or high-fat, high-cholesterol diet, it would also affect TM6SF2 expression. So as you can see from the immunostaining, so the expression of TM6SF2 is reduced when you fed the mice with unhealthy diet. Now, this is the key result of our study. So now we generated intestinal specific knockout of TM6SF2. So now TM6SF2 is not expressed in the intestine, but the expression is normal in the other organs, including the liver. So as you can see, this is very interesting. If you just look at the histology, the degree of steatosis, if you look at it very carefully using all red Osting, which is a very sensitive way of detecting hepatic steatosis, it is still increased, but not much. So if you look at the H&E, you really do not really see a marked increase in hepatic steatosis. However, on the other hand, you can see that inflammation increased. So as you can see, both for the ALT level, for T-bars, the marker of oxidative stress, as well as the inflammatory cytokines, TNF-alpha and the downstream interleukin-6, all those were significantly increased. So at the same time, NF-kappa B, which is central in the pathogenesis of steatohepatitis, is upregulated, and with a corresponding downregulation of I-kappa B alpha. And at the same time, we also performed flow cytometry to show that the knockout of TM6SF2 in the intestine would reduce the number of M1 macrophages in the liver. So M1 is the pro-inflammatory type of macrophages, so as shown on the left-hand side. So when we look at the different pathway changes. Then after knocking out the intestinal TM6SF2, the pathways that are most affected would include some cytokine-mediated pathways, lipid metabolic process, and importantly, inflammation and also leukocyte chemotaxis. So the attraction of white cells to the gut lumen. So these are all related to inflammation. Okay, so now we fed the mice. So just now when we showed that the knockout mice did not have marked increase in hepatic steatosis, we were feeding the animals with chow diet. And now we fed those mice with choline deficient high-fat diet. So as you can see here, then it hit everything. So not only did hepatic steatosis increase markedly, at the same time, there was also significant inflammation. So recapitulating all the features of steatohepatitis. So our first hypothesis was that this is partly driven by endotoxemia. So this may be due to leaky gut, whatever. So this has been confirmed in this model. So in the intestinal knockout mice, indeed, in the circulation, you can see increased lipopolysaccharide, that is the bacterial endotoxin, which will stimulate toll-like receptor 4 and therefore inflammation. So not only in the circulation, we also found increased endotoxin inside the liver and also in the portal vein. So it completes our hypothesis. So it is generated in the gut, going through the portal vein, and finally to the liver, generating inflammation. So why would there be endotoxin in the liver? Then, of course, it is at least in part due to gut permeability. So we did experiment to test that, and it was positive. So long story short, we did both in vitro and in vivo experiments using labeled dextran. So we fed the mice with dextran and then 4 hours later to measure the level in the circulation. So an increase in the level indicates that there is increased absorption due to gut permeability. More than that, when we performed electron microscopy of the intestine of the mice, we saw basically obvious disruption of the tight junctions. So, so both in terms of morphological examination and also in terms of measurement of the tight junction width. So at the same time, the adherent junction protein E-cadherin is also diminished in mice with TM6SF2 knockout in the intestines. Now, gut permeability is one thing. The other thing that is tightly related would be, of course, gut microbiota dysbiosis. So this is the principal component analysis, clearly indicating that by knocking out TM6SF2 in the intestine, you would change the gut microbiota composition. And then we did metagenomic analysis and show that the harmful bacteria are increased, including, for example, Porphyria monodecia. But some of the beneficial bacteria, several strains of Ruminococcaceae were downregulated. Okay, and untargeted metabolomic profiling further identified lysophosphatidylate acid, or LPA, as a key enriched metabolite in those animals. So both in the stool, but also in the portal vein and also liver tissue, again completing the gut-liver axis. So we believe that this metabolite is produced by the bacteria. And in fact, by integrative analysis, we confirmed the association between the production of LPA and also some of the top identified bacteria. So in particular, the bacteria shown in red produced the corresponding enzymes responsible for the production of LPA. In order to confirm that LPA indeed is responsible for the development of inflammation, we directly applied LPA, and indeed in vitro models using the alpha mouse liver cells. So you can see that by adding LPA to the medium, we can increase the degree of both steatosis and inflammation. So again, both the inflammatory cytokines, TNF-alpha, IL-6 were upregulated, and also there is marked increase in the ALT and AST in the medium. So we want to know what other genes are affected if you knock out TM6SF2 in the intestine. So this time we performed a gene expression analysis. So we identified actually, we identified many changes, but the two main affected genes, so those would be highly upregulated, would include fatty acid binding protein, FABP1, and also pyruvate dehydrogenase kinase 4. So those are responsible for the production of lipids. Okay, so because of the upregulation of these genes, so one effect is that not only do we have fatty liver, we also have fatty intestines. So this is what we saw. So in the intestine of the knockout animals, you can see fat accumulation in the epithelium of the small intestine as well. So the increased production of free fatty acids in the small intestine cells of TM6SF2 is further validated. So this time we performed a lipidomic analysis to look at the changes. So we identified several free fatty acids that are particularly upregulated. And the most dominant one is actually arachidic acid. So arachidic acid and arachidonic acid in isolation when added to our transwell experiment would increase gut permeability. So we think that this is part and parcel of the effects of LPA and also gut microbiota changes. However, upon further analysis, only arachidic acid was associated with increased LPA in the portal vein. So we think this is the main pathway being involved. In order to demonstrate that this is a causal relationship and not just correlation in nature, we performed fecal microbiota transplantation. So we moved the stool from the knockout animals to germ-free mice. So this is in collaboration with China Science. So we show that just by transplanting the unhealthy feces to those germ-free mice, you would be able to induce steatohepatitis. So you can see both increased fat here and also inflammation. And also, in terms of the therapeutic side, so whether we can do something about that, so we are still working on that. Theoretically, there are other things we can do to manipulate that, including the use of probiotics, prebiotics, or symbiotics. But here, we had a very simple experiments. We co-host the knockout animals with wild-type mice. So theoretically, the wild-type mice would contain the healthy gut microbiota, whereas the knockout animals would contain the unhealthy microbiota. So we host them together for 4 months. So as you can see from the experimental design, so the animals lived for 12 months in total, but by the 8th month, we co-host them. And therefore, theoretically, there would be exchange in gut microbiota among the animals. And very interestingly, just by this kind of arrangement, we were able to demonstrate that the knockout animals no longer had hepatic steatosis or inflammation. And if you check the inflammatory cytokines and also markers of oxidative stress, you can also see that would be largely suppressed. So on top of that, we also show that the co-host animals, so even though the TM6SF2 was knocked out, there would also be reduced macrophage activation. And on top of that, when we did EM analysis, you can also see that even the intestinal tight junction has been preserved. So now you see healthy tight junction, and therefore the gut is no longer leaky. So when you measure the tight junction width, it is normal. And therefore, in conclusion, Chairman, ladies and gentlemen, in a series of experiments, we demonstrated that TM6SF2 in the intestine actually matters. So in the intestine, knockout or suppression of TM6SF2 would result in an increase in the metabolite arachidic acid, which would in turn lead to gut microbiota dysbiosis. This would in turn affect the gut barrier dysfunction, and also the unhealthy gut microbiota would produce the lysophosphatidyl acid, which is harmful. And together into the liver, it would lead to lipid accumulation. It would lead to inflammation, so a key feature of steatohepatitis through upregulation of NF-kappa B, inflammatory cytokines, and finally to steatohepatitis. But of course, we shouldn't forget that TM6SF2 inside the liver is is also important because it contributes to the accumulation of fat inside the liver. And with that, I thank you very much.
Thank you, Dr. Wang. So nice lecture for you comment. Okay.
Yeah, thank you for your great talk. My name is Kazuki Kasa from NTU Singapore. So my question is, what kind of bacteria can produce LPA in your system?
Okay, so in our experiment, there are 4 bacteria that are upregulated. So among them, 2, I think one would be bacteria. So there are others. So as illustrated in our metagenomic analysis. Yes, and a functional analysis has been performed to show that they produced the corresponding enzymes responsible for LPA production.
Okay, next, please.
Stefan Stender from Copenhagen. Great talk. I have a question about the Human Genetic Association, where you showed an increase in the homozygotes.
Yes.
Was it LPA or the marker of?
So in that study, actually, we tested 4 endotoxemia biomarker. What I showed was a lipopolysaccharide binding protein, which would be an indirect marker of endotoxemia.
Question is, do you think it's a recessive effect, because there didn't seem to be anything on the heterozygotes?
Yes, very interesting. So as you can see, for the wild type and also the heterozygous, they were essentially the same, not only for this marker, but for all the other endotoxemia biomarker. So I think you really need to be homozygous when you lost all the function of TM6SF2 in the intestine. Would you start to have leaky gut and also endotoxemia?
Okay, Dr. Nishikawa, please.
Thank you, excellent presentation. My opinion is endotoxin-producing bacteria increased only in intestine-specific knockout mouse. What about liver-specific or systemic knockout mouse?
This is a great question. We didn't look into that. I would envision that for the global knockout mice, there would be corresponding changes. For the liver-specific knockout animals, I expect, because of the crosstalk between the gut and the liver, there may be changes as well, but unlikely to be identical. But this is a great question. We should have looked into that as well. And since we have the animals, this can be done.
Okay, I understood.
Thank you. So please later we'll discuss more.
Thank you very much.
Thank you. And third presenter is Dr. Jin Kao Fan. So, however, unfortunately, we cannot find out the Dr. Jin Kao Fan. So, is there any presenter on behalf of Dr. Jin Kao Fan is here? No.
Dr. Shinji Tanaka. Dr. Shinji Tanaka, a professor of the Tokyo Medical and Dental University, is busy with operation, so he cannot attend this meeting, and he cannot send message or attend the web. So we have to skip him. So whereas 4 papers will be presented in this session, only the 2 persons can be presented. So as I first mentioned, the 2 persons come to the
On this stage,
on this stage, and we'll have the discussion time. We have much time, so many questions can be made.
And Dr. Tanaka, Professor Tanaka said so he sent a video lecture. However, technically we cannot receive the video message. So unfortunately we skipped it and Professor Tanaka's lecture.
Many audience have some questions to the 2 persons. The first is the Professor Kawaguchi, who talked about SCOT2 inhibitor, the beneficial effect of these drugs for the NAFLD and other types of diseases. The some audience have good discussion.
So first,
Dr. Nishika, please.
I would like to ask Professor Kawaguchi. Okay. My question is SGLT2 inhibitor associated with elimination of regulatory T cell? What do you think about this?
Thank you, Professor Nishika, for your great question. And I just want to make sure that your question is SGLT2 inhibitor eliminates the T cell in the liver.
Yeah.
I have no idea about it. That what I did show as an effect of SGLT2 inhibitor on the PDL1, but it's a CD8 positive T cell.
Yeah.
And the SGLT2 inhibitor promotes the degradation of the PDL1 on the hepatoma cell. That allows the attacking by the CD8 positive T cell. That may cause the suppression of SCC. But it's in vitro study, so we do not know about that occur in a patient with SCC.
Okay, thank you. I understand.
Thank you.
Understood.
may I ask a question for the, sorry, Dr. Hino.
I have a question for Professor Wang. Yeah, I'm very interested in the TM6SF2. And so do you have any idea about the difference of the phenotype in the liver-specific knockout mice and intestinal-specific knockout mice, especially the different occurring of inflammation between liver-specific and intestinal-specific knockout mice? So what determines such a difference in the liver and the small intestine?
Yes.
Do you have any idea about that?
Yes, this is an excellent question. So from what we have learned from other teams before, so we know that the function of TM6SF2 in the liver is mainly for the handling of the lipids, so for the lipidation and also release of VLDL. And therefore, if you cannot do that, the lipid will be trapped inside the liver. And therefore, it is mainly fat. But what our study showed was that this is insufficient to cause inflammation, but it is really inflammation that would eventually drive fibrosis, cirrhosis, and all sorts of complications. So I think the main value of this piece of information is that the intestine is extremely important in terms of steatohepatitis development. So you need the dysfunction of TM6SF2 in the intestine to generate the endotoxin and also the harmful metabolites that would go through the portal vein into the liver to induce inflammation and tissue injury. And together with the accumulation of toxic lipid species inside the liver, that is how we develop steatohepatitis. So I think the study is interesting in dissecting between the function and also the pathophysiology of the TM6SF2 in different organs. I have a question for Professor Kawaguchi. So, yes, excellent talk, very interesting data. So, now if the SGLT2 inhibitor function converge towards also AMPK upregulation. So, have you ever thought about whether if you combine metformin, which would also upregulate AMPK function, together with SGLT2 inhibition, there may be synergistic effects on both steatohepatitis and also even HCC prevention?
That's a really good question, Professor Vincent. And actually, we've been good friends for a long time. And thank you for your kind question. And there is one important paper published in last year. They investigated the additional effects of SGLT2 inhibitor on the patient treated with metformin who were in the diabetic cirrhosis. They shows that by addition of SGLT2 inhibitor, there is a significant improvement 5 years overall survival and a 5-year decompensation with liver. And surprisingly, the most impacted factor was a suppression of onset of HCC. So I guess it's almost the same, but upregulation of AMP kinase, but metformin and a SGLT2 inhibitor combination may work synergetically to suppress the liver disease, I guess. And I have a question to the from the audience, please. First, please.
Thank you very much. My question is directed at Professor Wong. My name is Han Kiet Ho from the National University of Singapore. So with regards to the polymorphism of TM6SF2, what do we know more information about the prevalence of the polymorphism, especially if there is any subgroup analysis that may compare the obese and the non-obese type of MAFLD patients?
Okay, excellent question. So first of all, unfortunately, this is not a common polymorphism. So in any population, it accounts for less than 5% of the general population. However, recently, together with Terry Yip in the audience, we have analyzed the 1,000 Genome Project, and we realized that actually, around the globe, East Asia would have the highest prevalence of this gene polymorphism. So, that in a way, this may explain why fatty liver disease is still quite common in this part of the world, despite the fact that we are not that metabolically burdened. Sorry, the second question is?
One question, if I can ask Professor Kawaguchi another question. Yeah. SGLT2 inhibitors, there are a few that are already commonly used in the market. I'm just wondering whether there is any comparative study to show that the efficacy of what you have described here correlates to the potency of the SGLT2 inhibitors.
Thank you for your question. Your question is there any different cross effects of SGLT2 inhibitor? That's a difference is a maker, right? As far as I investigated, there is no so much difference among the drug. The most reported SGLT2 inhibitor is the canagliflozin. That is reported as on like anti-tumor effects as well as on anti-diabetic effects. Thank you.
Sorry, I remember the second question now. So you were asking about the gene polymorphism in obese and lean individuals. In fact, this is something we looked into. So what we found was that the relative importance of PNPLA3, the most important gene, would be higher in lean individuals in conducing fatty liver disease. However, there was no differential effect between obese and lean individuals for TM6SF2 mutation.
I have a question to Vincent. Is it okay? Yeah. And I was so impressed by your presentation. TM6SF2 was very involved in the leaky gut. So I was wondering, is there any diseases that is involved in the TM6SF2 rather than liver disease, such as like an IBD or allergic disease or autoimmune disease, because gut microbiota is involved in not only liver disease, but also many diseases.
Okay, for IBD and autoimmune diseases, I'm not aware of the data, but paradoxically, the presence of TM6SF2 mutation would reduce cardiovascular risk. This is because if the lipid is trapped inside the liver, the circulating lipid level would be reduced. So in the long run, those individuals would have a lower risk of myocardial infarction and ischemic stroke. And therefore, it is not a good therapeutic target. So we just want to work out the mechanism and identify how we can manipulate the changes to improve the treatment of steatohepatitis.
Thank you.
I have a question to Professor Wang. You showed that the TM6SF2 expression is high in liver and small intestine and kidney.
Yes.
What would be the kidney if the knockout is made?
Oh, the simple answer is I don't know, but from the physiological studies that we have learned from both the intestine and the liver, it is likely to be generate the handling of lipids. And therefore, in any organ, if you downregulate that, it is likely that would lead to fat accumulation. However, the pathological effect in the kidneys may not be as important as compared with the other two organs.
Thank you. And I have one question okay, before. And one more question. We have the data that more than 1,000 patient, MASH patient in Japanese. We studied the variant, non-function variant of PNPL3, HSD 13 beta, and TM6SF2, 3 genes. So in the longitudinal study, we know that the hepatic decompensation HCC, hepatic event is confirmed in PNPL3 and TM6SF2, 2 genes. So we also wanted to know if the cardiovascular event is changed in the TM6SF2 variant. So we cannot see such event in our population. Then that may be caused partially by the lean MASH patients is frequent in the ASEAN. Do you agree with that comment?
I wouldn't be too surprised because after all, for any metabolic diseases or cardiometabolic diseases, we are usually talking about multiple genes being involved. And also at the same time, there is gene environmental interaction. And therefore, a single gene is often insufficient to drive everything. So, so in your study, I think it is also important to study the environmental part. So the dietary, lifestyle, and other things. So because Japanese are so healthy, so a single TM6SF2 may not be sufficient to change or put it this way, if the incidence of cardiovascular event is quite low, then it would be very difficult for a candidate gene study to find out the change.
Thank you for your next question.
I have a question for Professor Wang. It's about the plasma lipid phenotypes. Do you see any difference in the liver-specific TM6 knockout mice and the intestine-specific knockout mice in their plasma lipid phenotype?
We didn't look at that. So we only look at the lipidomics and metabolomics in the plasma and also in other tissues of the intestinal knockout mice. But it would be very interesting to compare.
I want to ask Professor Wang. I want to ask with regard to this biosis. And my question is, what is alpha diversity in intestine-specific TM6SF2 knockout mouse? Do you have any data about it?
I didn't show the data. There was no statistical significance in alpha diversity.
No significant increase?
We didn't demonstrate that. And actually, even in our human studies, so I know that there is a lot of heterogeneity in the studies. So even in our human studies, we were not able to demonstrate differences in alpha diversity between people with MAFLD compared with those without.
Okay, thank you.
May I ask to Dr. Wang from me? So you mentioned the intestinal specific knockout mice increase the arachidic acid in the intestine. And you also mentioned the intestine in the lipid accumulation was increasing, is it right?
Yes.
So is there any data of this? So those morphoformist patient also develop the intestinal steatosis in the human data?
Human data, I'm not aware of. However, in both a zebrafish model and also mouse model, this has been repeatedly demonstrated. So accumulation of fat goblets inside the epithelium. But humans, unfortunately, it is expressed mostly in the small intestine. So if you do an endoscopy study, you have to go very deep before you can get the proper tissue.
And next question is, so it's very interesting. The co-hosting is reducing the impairment of the so dysbiosis. However, also the knockout mice is increased arachidonic acid in the intestine. So, however, so only co-hosting did not change the arachidonic acid increasing in the intestine knockout mice, I think. So do you think why some dysbiosis is improvement by just co-hosting without the change of the arachidonic acid in the level of the
Okay,
intestine?
I would say that the metabolite is only one thing. So when you talk about gut microbiota changes, it is often with changes in multiple metabolites, including short-chain fatty acids, for example. And therefore, this by itself is not sufficient to explain everything. However, whenever you have a co-housing experiment indicating changes in the phenotype and the disease status, it has to be due to exchanges in the gut microbiota. There are no other explanations unless one mouse is biting the other.
So just a co-hosting is also improvement of the gut leak is also improvement by co-hosting.
But the leakiness has to be through the microbiota changes.
Okay, thank you.
Yeah, I'm Hayato Nakaga from university. I'm sorry for delay, and I couldn't see the presentation of Professor Kawaguchi. I'm so sorry. I would like to ask a question for Dr. Wong, and regarding with Professor Hikita's question, if knockout of TM6SF2 in intestinal epithelial cells is still remained, why just co-housing improves the phenotype? I can't understand.
Yes, I think this is only very preliminary, so I don't think that would be the ultimate answer. So, but what we are working on would be to try different probiotics and prebiotics to see the changes. So, because the genetic changes inside the intestine is something you cannot modify. So we have to think about things that we can modify. And gut microbiota is the most logical target. So this is what we are working on now.
Do you have any data for human?
Human, you mean
microbiota?
We do, but not exactly through the interaction with TM6SF2. I think the main trouble for us and also for most groups, I believe, is that the homozygous prevalence is so low, and therefore you will have limited statistical power to delineate that.
I see. And another question is that, as you said, TM6SF2KK is known to reduce cardiovascular risk. So did you check the effect of intestinal knockout of TM6SF2 on cardiovascular systems?
Interesting. I didn't think about that because I thought that the reason why it would affect the lipid level in the circulation is mainly because of the liver effect. So I sincerely haven't thought about that.
Because based on your concept, maybe systemic inflammation may be induced by intestinal knockout of TM6SF2.
Yes, that is true.
Yeah,
because inflammation itself is known to be pro-atherogenic.
That may be rather bad for cardiovascular system.
Exactly.
Okay.
A question for Professor Kawaguchi. The protective effect of SGLT2 inhibition, was that proportional to the glucose lowering effect you see? How much of the protection do you think is mediated through that?
Thank you for a very important question. I think it's a parallel, but the mechanism is independent. So if you see the patient who do not respond to the SGLT2 inhibitor for the glucose lowering effects, I think SGLT2 can be expected to suppress the HCC.
Along the same line, we can also ask, how about weight reduction? So how much do you think weight reduction contributed to your observations?
Thank you for your question. I think it's the contribution of the body weight reduction is, I think, little, because it's not so big reduction in the body weight by the SGLT2 inhibitor. Thank you.
So Professor Tanaka newly uploaded his presentation data, so now we coming received. So if any another question, so please, thank you so much. Nice discussion. So thank you. And the next presenter is the Professor Tanaka. So Please slide start.
Thank you for giving me the opportunity to present our studies on metabolic dysfunction associated liver cancer. I have no COI to disclose. Heterogeneity is one of the hallmarks of cancers, including hepatocellular carcinoma SCC. Using surgically rejected samples, we have identified several molecular and immunological subtypes, such as metabolic syndrome associated SCC. This report selected as a highly cited paper placed in the top 1%. Interestingly, the metabolic subtype of SCC is not associated with specific genetic mutations, and so in this study, we applied the epigenetic approach to clarify the molecular mechanism of metabolic SCC. In addition to clinical samples, the mouse model of MAFLD MASH SCC was used to identify the common pathways for human and mouse metabolic hepatocarcinosis by cross-species analysis. First, we identified the epigenetic abnormality specific for human and mouse SCC with metabolic disorders. Using our datasets, we analyzed the expression levels epigenetic regulator genes between human and mouse SCC with metabolic disorders. As shown in the Vulcan plots, KDM6B was commonly down-regulated in metabolic SCC at mRNA levels as well as protein levels in the cancer cells of SCC. Previous studies revealed that 5-HMSC level is impaired in MAFLD tissues and diabetic patients, and it might be associated with relation gene expression. Then we analyzed the relationship between KD6B expression and 5-HMSC levels in human SCC lines. As shown, KD6B expression correlates with 5-HMSC levels at the CPG island within exons 70 to 18, suggesting that the regulation of KD6B expression may depend on the 5-FTC levels. Second, we analyze the role of KD6B downregulation. Histone methylation is one of the epigenetic system for activation or inactivation of gene expression. KD6B is a histone modifying enzyme of H3K27 demethylase that function as a transcriptional activator. To clarify the role of KD6B dysfunction, we established a KD6B knockout subclone from human SC cells using a CRISPR-Cas9 system. The genome-wide expression analysis revealed that KD6B knockout downregulates lipid metabolism-related genes involved the cholesterol and fatty acid metabolism pathways. Then, metabolome analysis was performed for comprehensive measurement of lipid metabolites. In KD6B knockout SC cells, intracellular triglyceride metabolism was markedly changed, especially after palmitate treatment. KD6B knockout reduced intracellular triglyceride levels by LC-ESI MSM analysis, as well as by validation analysis for direct lipid quantitation. Next, we investigated the biological effects after palmitate treatment. Oil red staining showed lipid accumulation was reduced in KD6B knockout cells. In addition, KD6B knockout increased significant resistance to lipotoxicity after palmitate treatment. These data were confirmed by siRNA analysis in AC cells. KD6B knockdown reduced lipid accumulation and increased resistance to lipotoxicity with downregulation of lipid metabolism-related genes ABAT, ACSL1, and G0S2. Next, we identify the target pathway of KDM6B downregulation. To clarify how KD6B regulates downstream target genes expression, we quantified KD6B occupancy and H3K27 methylation levels at the promoter region of lipid metabolism-related genes between KDM6B wild type and knockout cells. CHIP assays demonstrated that KD6B can bind the promoter regions of ACSL1 and G0S2 and induce H3K27 demethylation, resulting in positive regulation of this gene expression. ACSL1, adipose acetyl-CoA synthase 1, is an enzyme directing fatty acids towards mitochondrial beta oxidation. We analyze the effects of ACSL1 siRNA and that knockdown can reduce lipid accumulation, but not induce the resistance to lipotoxicity, indicating possibly not a direct therapeutic target in metabolic SSC. G0S2, initially identified as a G0S1 switch protein 2 in lymphocytes, and also function as an inhibitor of ATGL PNPL2 activity and regulates adipose lipolysis. Knockdown of G0S2 resulted in reduced lipid accumulation as well as increased resistance to lipotoxicity, similar to the effect of KD6B knockout or knockdown. These results suggested G0S2 as a potential target pathway in metabolic SCC with KD6B downregulation. The significance of G0S2 pathway was validated by reverse examination. In KD6B knockout cells, G0S2 overexpression increased intracellular lipid accumulation and reduced resistance to lipotoxicity. These results confirm the mechanism of decreased KD6B expression contributes to acquisition of resistance to lipotoxicity via decreased G0S2 expression, suggesting the therapeutic targets. Indeed, as a direct target, ATGL activity was significantly upregulated in KD6B knockout SC cells and G0S2 knockdown SC cells. Then we evaluated the effect of ATGL knockdown or treatment of ATGL inhibitor atoglistatin. As shown, inhibition ATGL pathway increased lipid accumulation and suppressed the proliferation of KD6B knockout SC cells. However, in KD6B wild-type HC cells, the inhibition of ATG pathway shows no effect on lipid accumulation and cell proliferation. These data strongly suggested that ATGL-PNM2 is a specific pathway for treatment as well as prevention of metabolic subtype of HCC with KDM6B downregulation. Finally, clinical significance of KD6B downregulation was evaluated in HCC with metabolic disorders. According to the immunohistochemical analysis using our surgically resected samples, KD6B downregulation was identified as one of the characteristic findings of MAFLD SCC. In addition, correlated downregulation of KD6B G02 was recognized in MAFLD SCC. Our studies indicate that targeting ATGL-PNM2 pathway may be an effective treatment of the metabolic subtype SCC. Previous studies revealed that PNPL3 SNP variant can function as a competitor of ATGL-PNM2, leading to lipotoxic steatosis in hepatocytes. Our studies revealed that ATGL-PNM2 activation is recovered KD6B knockout cells via suppressing G0S2 expression. As therapeutic targets against resistance to lipotoxicity, we will focus on ATGL-PNM2 and also H3K27 methyltransferase EZA2 for MAFLD SCC in the future. This is a summary figure. MAFLD SCC shows frequent loss of KDM6B. Loss of KDM6B epigenetically confers resistance to lipotoxicity via ATGL-PNM2 activation by G0S2 suppression. Targeting KDM6B G0S2 ATGL pathway may be an effective treatment for MAFLD SCC. My collaborators are shown here. Thank you for your attention.
So this is video presentation, so we cannot receive questions. So thank you for joining this session and active discussion from audience. And there are some troubles in this session, including me, but we could learn many things from 3 experts. And I think importance of MAFLD, MAFLD will be more increased in the future. But recently, new drug targeting THR beta resmetirom is just approved in FDA. This is very promising, but the treatment way is very limited. So further investigation about the molecular mechanisms of MAFLD will be more important, and hopefully important evidence will be launched from Asia. So thank you again for joining this session. It's close to time.
Thank you very much, the moderators, discussant, and all speakers.